Welcome everybody to 52 Living Ideas. I am delighted to have Agastya Sharma here to talk about the gut microbiome and its impact on our health. Now, Agastya is one of my oldest friends, though he's only 25 because I've known him since he was 1. And he's been a great friend throughout the time. So it's just an honor to have him here. Welcome, Agastya.
Thank you, Shrikant. It's great to be here as well.
Wonderful. So why is it that there is so much talk about the gut microbiome? Why?
Sure. So first off, just to kind of introduce like the idea of kind of the gut and the human microbiome. When I say like the gut or human microbiome, that just refers to all of the species of bacteria, fungi, and archaea and viruses that live on or inside the human body. So one of the key scientific innovations that kind of has driven recent interest in the gut microbiome is actually DNA sequencing. So looking at kind of the bacteria around us, for centuries now, we've been able to culture bacteria and grow it outside of its system on petri dishes. But the problem with that is a lot of bacteria rely on their ecosystem and their environment to survive. So if you pull it out of its environment, it's not going to be able to survive and you can't study it. And it's estimated that about 90% of the bacterial species that are present in the world cannot be cultured on single culture plates, which is a problem for studying. However, I'm just going to show a quick chart to kind of drive this point home. But recently what's been happening is people have been looking at classifying life by DNA sequencing and analyzing how similar species are by looking at their the percent similarity of their genomes. And the reason we're able to do this is because of this chart, or the information that this chart shows here, is in 2001, to sequence 1 million bases of DNA cost about $10,000. And currently it costs about 1 cent. So
Wow.
this kind of this kind of innovation basically means that it's really, really, really cheap to start sequencing DNA. And so now what people are able to do is they're able to go in and just isolate all of the DNA from a given system, like let's say from the feces of an animal or from an organ, and then sequence all of it. And what this has kind of led us to find is that there are a lot of bacterial species that we didn't know about that are key drivers of human health and are key players in the gut microbiome that we're finally now able to study simply because we know they exist. So in the past 10 to 15 years, the gut microbiome has a large reason of why people are interested in the gut microbiome is because we didn't know about it 20 years ago.
Wonderful. Let me ask about you personally. And I know your science teacher is in the audience. Why did you get interested in this particular topic?
So I got interested in microbiology in general through actually when I was learning about infectious diseases in high school. And I was just amazed at how something that's so small that you can't even see it can just wreak absolute havoc on human health. And then so that led me to University of Wisconsin, where I got a bachelor's in microbiology in May of 2020. And while I was there, I was working on parasitology, and then kind of that drove my interest in working in microbiology in graduate school and later on in my career. And so now I'm at Duke University, where I'm a graduate student in the Department of Molecular Genetics and Microbiology. So I'm pursuing my PhD. And when I got to Duke, I started looking at different bacteriology labs because, again, I was still interested in kind of this bacteriology. And I was introduced to Commensal Bacteria. So Commensal Bacteria are bacteria that live alongside or within us, but don't necessarily cause harm. There's a sort of mutualistic relationship that occurs between us and them where, we kind of have this safe environment that bacteria can flourish in, and in return, these bacteria help us survive. And I got really, really interested and fascinated by that. And that has sort of led me to studying the gut microbiome. And so just as a quick, I guess, couple of sentences, what I do is I focus on a single species of gut bacteria called Akkermansia. And Akkermansia, which you may or may not have heard of, it's kind of becoming more and more popular in the news, is a species of bacteria that survives by living within and eating intestinal mucus. So intestinal cells create this thick layer of mucus that separates the bacteria in the intestine from kind of infecting and damaging human tissue. And so Akkermansia is able to live within this and eat it up and degrade it. And what people have found kind of counterintuitively is that the more Akkermansia you have in your gut, the healthier you are in general, the better you're able to respond to cancer therapeutics if you have cancer. The leaner you are, the better BMI you have, and the less fat you accumulate through your diet. So that's a little bit about why I do gut microbiome science.
I mean, the thing, Agastya, is like, when I studied biology in school back in India, I hated it because it was all about remembering specific things. But when I started studying it on my own, reading Darwin and trying to figure out, it's actually very fascinating because each of these things are living things. Like, for example, the point that you're making is that the bacteria can exist only in the environment, certain environment, and they interact with each other. All these things interact with each other and in the environment. Like, for example, we're talking about gut microbiome. They interact with us, the health of the being that they are part of, in a very complex way, and to understand, kind of follow their life. So I really like the fact that you're focused on this one species, because you're kind of like saying, okay, what is the personality of this thing? What does it do? How does it function? What are its limits? Well, wonderful. So kind of stepping back, if you want to explain to someone, what is the function of the gut microbiome in human beings? What would you say?
So the gut microbiome is unique because it's a community of bacteria that's located at the intersection of a lot of organ systems within the human body. So the gut itself obviously is involved in digestion and the digestive system. So right away, we can say that the gut microbiome has a huge role in controlling controlling metabolism in general. In fact, there's this, I guess, I mean, we can also get into it later, but there are interesting studies that show that if you take the gut microbiome of obese mice and transplant it into lean mice, then the lean mice start to become obese without changing their diet at all. So that's one example. The gut is also very, very, very connected to the brain through the vagus nerve. And so that is a major influential connection that allows the gut microbiota to somewhat influence nervous system and brain activity. And indeed, people have found that gut microbiomes, because they're so diverse and complex, tend to be very variable from person to person, even between twins. But what people have found is that the gut microbiomes of people who have autism or people who have schizophrenia are very, very similar to each other, indicating that perhaps there is some sort of relationship between kind of the gut microbiome and our brain activity.
Wow, this is fascinating. So what you're saying is that the general kind of health aspects that we observe with human beings at a macro level of the weight that they have, what is going on in their brain, is connected with, there is a causality. I don't know, one way, both ways, I don't know, but there is a causality between that and the nature of the gut microbiome. Wow. So let's look at that further. So because that's something like, for example, 30 years ago, 40 years ago, people would not have known about this at all, right?
Yeah.
Go ahead.
No, I was also going to say, just to kind of hammer home a little bit more the point of kind of how amazing and intricate the gut microbiome is, the human body is made up of approximately, I'd say around, I think it's 3 to 10 trillion human cells. However, the human microbiome itself is made up of 10 to 100 trillion bacterial cells and archaeal cells and viruses. And it's such a vast community that that allows us to kind of survive. And it's estimated that there are about 3 million unique microbial genes that are constantly expressed in the human gut, just between all of these bacteria. For example, there are proteins called, I think they're, it's trypsin proteins that help us digest proteins. So they go through, and once proteins are, they basically break up these long chains of proteins into single amino acids. And the human body encodes something on the order of 15 to 20 different protease, or these kind of protease enzymes. Whereas the gut microbiome has hundreds, if not thousands, of unique proteases that are just designed to break up specific kind of protein motifs. And without those, we'd be severely impaired in the amount of food that we could digest. So it's
Wonderful.
It gets to, yeah, no, sorry.
Go ahead. It gets to, go ahead.
I was just gonna say, it gets to a point where you wonder if humans could, I guess, even survive in the way we do without these bacteria.
Yeah, absolutely. So firstly, I want to say that it's great that it's not a democracy, because it looks like otherwise we would be outvoted. Our cells, human cells, would be outvoted.
Yeah.
So, and that's the thing about biological systems, that they exist in a hierarchy, in a certain structure, and you want to understand the structure. So the fact that there are more gut microbial cells in us than human cells, I mean, that is stunning. First time I heard it, it is, and it's still stunning to me. Now, let's look at the variety. So let's talk a little bit about variety. So gut microbiome, how many different species of bacteria are we talking about?
So I have a chart, or a tree of life, to kind of hammer this point home a little bit about how diverse.
Okay, let me interrupt you for a second. See, I'm used to going back and forth with you, so sorry to keep interrupting you. But I want to make a point, folks, because what happens is that science is important, and science is transforming our lives. Unfortunately, a lot of the science that happens, which actually is impacting our life, happens in these labs and in universities. And most people who are doing science actually are not, many of them are not very good. There are some exceptional exceptions, like, Richard Feynman, who can bring, or even Albert Einstein, okay? They can make their thoughts clear to anybody. But science and technology is transforming our life, and we need to understand it. We need to make efforts to understand it. And people who are doing science and technology actually depend on the support from everybody else in order to carry out their ideas, not only to produce the ideas, but to, take those ideas and transform civilization. So this communication on science, of the deepest science that is taking place, is of great value. So I am delighted, Agastya, that you write a blog. So tell me a little bit about why you're writing a blog, and what do you think about this point about communication?
Sure. So I definitely think that, I guess one of the things that I started noticing the more and more I got into science is that, we're doing all this groundbreaking work, and within universities, every university has like so many different seminars and talks and conferences. And if you're a scientist, those are amazing to go to, and just to kind of learn more about science. But when you look at how science is presented to kind of the general public outside of scientific journals, you start to see that kind of information gets lost in translation. And one thing that kind of people like to see in news is this sense of conclusiveness. But the issue with that is that science is inherently inconclusive. And that's So that's kind of one of the main struggles that I've noticed with scientific communication. And that's one of the reasons why I started this blog, is because I really, really enjoy bacteriology. And there are so many different things that are happening in all different kind of all these different spaces, like biomanufacturing or bioengineering of new materials, or just in once again, kind of human health and infectious disease that are happening, that are advances in bacteriology that people don't really know too much about. And so as a way of kind of communicating these new innovations to the public, combined with, I guess, my own goal of improving myself as a scientific communicator, I started this blog to hopefully kind of disseminate some science and make it a little more accessible and relatable.
Wonderful. And I'm glad that you came on this 52 Living Ideas, because this is another way of doing it. Because what happens is that no matter how hard you try to put things into writing, it's only when you start, hearing people's questions, responding to them, you have
Exactly.
a far more kind of a holistic way of seeing where the other person is and what you need to say, and then you put that into your writing. So your writing becomes far more accessible to people. So I'm glad you're doing this.
Exactly. It's great to be here for that.
Wonderful. So please go ahead. You are about to show us a slide.
Oh, sure. Also, one other note that I will say, when you're mentioning Richard Feynman and Albert Einstein, I'd also like to say Carl Sagan is an amazing scientist and is very, very good at communicating ideas himself. So that's another model of kind of how to make science.
Yeah, just to follow up on it. Okay, we're going to have to keep going on these slight tangents. There is no other way. But so, for example, I remember listening to Carl Sagan. There was a center in Bombay called US. I don't know. It's like, it was basically a way for US government to counter Soviet propaganda. And the way it did it was to actually provide information about what was going on in the West. And when Cosmos came out, I think a few years after that, they played Cosmos in this large theater. So I still remember as a kid going and watching Cosmos and the effect it had on me. The phrase that still rings in my mind is billions and billions. He would keep saying that, but to tell you that the world is complex, there is a lot going on in the world. And a sense of awe with which he said it was very infectious. So I'm delighted you brought that up. Go ahead.
Okay. So yeah, Carl Sagan is an amazing orator. When you speak. But yes, so this is, I apologize if it's a bit grainy, but I'll kind of talk through it a little bit. So this is the bacterial tree of life. So looking at kind of archaea, eukaryotes, and then everything here is like all bacteria. And if kind of human beings and other animals would be plotted on the same tree, they would be about basically the branches would be no wider than the current eukaryotes. So just looking at this visually, you can appreciate how many distinct bacterial species there are in the world. And obviously not all of these are represented within the human gut, but a good portion of them are. And I was looking, I guess, to kind of drive this this kind of point home again. One of the key bacterial species that people have studied and people have gotten very sick from is E. coli. And looking at, there are two strains of E. coli that are as genetically identical as humans and chickens, and yet they're considered the same species. So just to kind of highlight how broad and diverse bacteria are. I mean, even if you consider like all of the animals that are alive now, all of the mammals have basically, and even actually all the birds and reptiles have like basically the same metabolism. You convert glucose into carbon dioxide and ATP, which is, if you're not familiar, is a molecule that transfers energy from food into other biological systems. But bacteria, so that's, I like to kind of say, animal metabolism is boring because it's all the same thing, regardless of how diverse the animal kingdom is. But bacteria are able to do that with glucose, with other sugars. They're able to take carbon dioxide and convert that back into sugars. Bacteria have even started living on radioactive waste. Bacteria have also started living on plastics and kind of working to degrade those. So it's just an incredible amount of bacterial diversity that we still don't completely understand. That's kind of driving the world that we live in.
If you want to put it in terms of numbers, I don't know whether you know, if you look at the biomass of all the bacteria and compare it to all the multicellular organisms, do you know the number offhand?
I don't know the number offhand, but I know that the biomass of all kind of bacterial and microbial life is roughly similar to the biomass of kind of larger animals.
Yep. Yep.
So,
No, that's huge. I mean, it just gives you the scale, because like whenever people think of life, they automatically think of mammals. That's their default way of looking at life. But life is not like that. It's actually built, like, for example, plankton and the role plankton play in the entire chain of life is huge. And most people don't know that. Go ahead.
So a couple things that there was actually one point that I wanted to bring up, but bacteria are within the human gut. This kind of ties back to scale. There are upwards of 10 to the 7th, which is what, 10 million? There are upwards of 10 million bacterial cells in the human gut per square milliliter of space, or per square millimeter of space in the intestine.
Wow.
It's probably, the animal gut, I think, is probably the densest bacterial population in the world. But that kind of gives you a sense of how dense it is. And also to kind of bring your point, to your point of how most of life can be tied back to like cyanobacteria and plankton in the water. Looking at looking at the Earth, even billions of years ago, the Earth looked like Yellowstone National Park. You have hot springs and these very harsh environments, and bacteria began thriving in those. And to this day, there are bacteria called thermophiles that live in near boiling water and can't survive outside of boiling water. And because of that, the atmosphere, because of kind of volcanic eruptions and meteorites and the environment of the Earth at that time, the atmosphere was full of gases that we today can't breathe: hydrogen sulfide, carbon dioxide, methane. And that's where kind of microbial diversity was born, is there were all these molecules that could be used. And the way that kind of these molecules work is you take electrons from one and transfer them to the other. So within that, you generate a voltage. And so you can think of these reactions as basically just the transfer of voltage back and forth. And that's how you can quantify the efficiency of microbial metabolism. But suddenly kind of these cyanobacteria came along, and they were very good at generating oxygen. And because that is one of the most electrochemically favorable reactions, is the generation of O2 gas. And that over the course of a few years in kind of geological time scale, the atmosphere became so oxygenated with these cyanobacteria cranking out tons and tons of O2 gas that all of these bacterial species were essentially killed off in an extinction event and kind of forced to retreat into the corners of the Earth. So like underground or deep water vents or Yellowstone National Park or the human gut, for example. And it's because of these plankton and these cyanobacteria that there was enough oxygen for multicellular organisms to rise. And that's basically why we're here.
Wow. Wow. So folks, I mean, just grasp this, okay? These little things, little cell things, not only are they a critical part of you. In order to understand your health, you need to understand what is going on with these little things in you, which outvote you, by the way. But this is a crucial part of the entire story of if you just wanted to understand what's going on with the Earth as a planet, these little things are transforming the atmosphere, changing the composition of the atmosphere, changing what kind of animals are possible as a result of it, and driving as a driving force in evolution. So just, I mean, that's the whole story. Okay. So Agastya, I mean, this is a very large topic. So I want to take like different paths through it. So
Sure.
next path I want to go through it is about the story of the gut microbiome of an individual human being. So how does this begin? You know,
Mhm.
we start from an embryo, and we become, and we have a long life, and then we die. Can you talk about the microbiome, the gut microbiome? What happens to it when, and what are the various stages
Sure.
throughout a human lifespan?
Sure. So the gut microbiome, you can kind of think of, I like to think of the gut microbiome in terms of an ecosystem. So you can think of it as a forest, for example. And when you look at a forest after a forest fire, you start, yes, everything dies and it's a barren landscape, but then you start to see common species, people who've studied kind of wildfires, you see these common species pop up each and every time, as these are kind of the founder species. And then they kind of create the soil that's necessary for secondary species like dry ass plants to start growing, and then bigger and bigger trees can come in. So you have this kind of sense of establishing a community, and that is kind of the main story of an infant. So there's still a lot of debate about whether or not the gut microbiome kind of starts in utero, like and is transferred kind of through nutrients that are transferred through the placenta or not. But one thing that can be said for certain is that babies that are born naturally versus babies that are born by C-section have different gut microbiomes. And so this kind of passage of microbes from the skin to from the skin into the gut is very important for infants. And that's actually one of the reasons why they encourage skin to skin contact kind of immediately post birth, is to kind of help transfer this microbiome. Because especially the skin microbiome, has like species that are necessary because they help protect against infection. So there's a staphylococcus species that lives on the skin that outcompetes and prevents Staphylococcus aureus, which causes MRSA infections, from sitting and causing infections on the skin, and generally keeps the skin kind of dry to prevent things from growing. Then the other interesting thing that I actually found this out very recently from a colleague who's working on this, but the gut microbiome of infants who are fed formula is different from those that are fed breast milk. And one of the reasons for this is that breast milk contains these big complex sugar molecules called milk oligosaccharides, and that formula doesn't contain. And in humans, I think it's like a broad range. There are like 40 or 50 different milk oligosaccharides. And what's interesting is, so as I mentioned, human metabolism is very simple. You take glucose or glycogen, which are just chains of glucose molecules together, break off a glucose, and then you can metabolize that. So these milk oligosaccharides aren't designed for that. So why are they there? And it turns out the reason that they are there is specifically to feed gut microbes. So these milk oligosaccharides are essential for kind of helping gut microbes establish themselves within a community. And so the development of the gut microbiome is key, because you want to establish.
Sorry, I just want to highlight one thing. This is a very beautiful example of the symbiosis. It's like this
Exactly,
systems,
yeah.
Evolving together. So there is something that the milk has, our milk has, to feed our gut microbiome. Go ahead.
Yeah. So in infants, I mean, we know that infants in general are very vulnerable. They can't hold their heads up and can barely roll over. But from an infection standpoint, they're extremely, extremely vulnerable. And one of the reasons is they kind of, they don't have like this established microbiota within them that helps them fight this off, in addition to an immune system. So the development or an establishment of a resident commensal community in the gut is essential. And actually, if,
let me interrupt. I'm sorry. I'm going to keep on interrupting.
yeah,
I want to make kind of these because you're making so many points, which I think most people need to grasp, because most people's kind of view of health is very narrow. I mean, partly it is kind of Western medicine that focuses on fixing problems. So one of the things that is coming out of it is that most people, when they think of bacteria, they're saying bad. Bacteria are bad. But you can see the crucial importance of bacteria in human life. The positive impact of it is humongous, and you can't understand human beings without that. So go ahead.
Yeah, no, that's a really good point, Shrikant. Thank you. And so kind of going back to what I was saying with like the forest fire analogy. So you have these first species. So the first major species is known as Bifidobacterium. And that kind of blooms in infant guts. But then that kind of, what you see is that kind of tapers out as children grow older. And their gut, as you grow older, your gut microbiome becomes more and more stable, because you've kind of established this resilient community. Also, as kind of a side, just an interesting side note, if people have ever heard that you shouldn't feed honey to infants, this is one of the reasons why, is because honey can sometimes have Clostridium difficile spores in it. And Clostridium difficile, if it doesn't have, if it comes into an infant's gut and an infant doesn't have the gut microbiome to kind of drive it out, then it settles in there and creates a C. diff infection, which is bad, because of that kind of C. diff outcompetes any other bacteria and really doesn't allow the kind of gut microbiome as an ecosystem to take root.
Wonderful.
But what people have found is that gut microbiomes become more and more stable as you grow older. And so it's kind of harder and harder to change that community. But one thing, and if you're okay with kind of segueing a bit into the human health aspect of it,
take it wherever you want to go. This is very good. Please go on.
Cool. Okay. So one of the big human health problems that we currently have today is in antibiotics, and I'm sure we've all heard about antibiotic resistant superbugs that are popping up here and there. And one of the problems with antibiotics that are not directly targeted towards the species that they try to, or towards kind of an infectious species, is that they also kill off resident commensal microbiota. And that becomes a problem, because that's just kind of this massive disturbance of the gut ecosystem. And by killing off a lot of these key commensal organisms, you start to influence the growth of organisms that are kind of parasitic, or that are basically bad. And what's interesting is that those organisms are always there in the gut. It's just they're kept at a very, very low level. So what people have found is, for, I guess this kind of a tangential story, but it does bring my point home, is like there are species of Neisseria, which one species of Neisseria is what causes gonorrhea. But there are species of Neisseria that if you co-culture them with gonorrhea, they basically go and kind of create these enzymes that go and chew up gonorrhea DNA, preventing that bacteria from kind of taking hold. And similar things happen. I think I mentioned staph epidermidis kind of keeps Staphylococcus aureus at check. And similar things happen in the gut, where you have like lots of E. coli, for example, that create all of the kind of protease enzymes that I talked about before. But they also create these toxins that target other specific E. coli, for example, EHEC, which causes like enterohemorrhagic E. coli, or ETEC, from kind of popping up and causing problems within the gut.
Wonderful. I want to just follow up on one aspect here.
Yes.
That most Western medicine historically has taken almost like a billiard ball kind of a model of, from where they're saying, okay, there is a problem. Let me give you this that will kill the symptoms. So any antibiotics, kind of general antibiotics that just wipe out bacteria as such, is a great example of that, because
Yeah.
it's a living thing. Human body is a living thing. It's not just about fixing the particular problem that you're faced with, but to recover health. And health is far more complex, so kind of more targeted antibiotics. So that's a great example of antibiotics just wiping out this entire ecosystem that has been built over a long period of time, very slowly, very carefully. It has a very delicate balance. You're talking about bad things being there, but being held in check by other things. So it's a very complex system, and not understanding that system, just trying to solve one particular problem. Uh, and I see that as a general, and I want to give a recommendation to everybody. There is a fantastic book called Why We Get Sick.
Yes, I was going to look at that.
Yes, please. So go ahead. You can talk about it. Can you talk about the book? Because that's one of my favorite books. I think, your dad and mom is one of the people I gave this to. I just bought a whole bunch of books. First time I saw the book. I mean, I'll say a little bit about it and hand it over to you. It's a collaboration between George C. Williams, who's one of the greatest evolutionary biologists, and what's his name? I forget the name of the other gentleman who's a doctor who actually came on our program, 52 Living Ideas, to talk about applying the same idea. The core idea of the book is that health, the medicine, has developed independent of evolutionary theory. I mean, it's very odd. First time I heard it, I said, what? Yes, but it's true. It has developed separately. And what this book does is that it shows you that if you understand evolution, you understand many things about health. And a lot of the problems of health come from not understanding our evolutionary heritage. And the book started this entire field of evolutionary medicine. So it's very, go ahead.
I mean,
what do you do?
I don't have, I guess, too much to add beyond what Shrikant had said. But I think it's very good at kind of breaking from a communication perspective. It's very good at kind of talking about, as Shrikant said, why understanding this kind of evolution is important. And that's one thing that I actually touch on a lot in my blog, is like whenever I'm looking at different systems. So for example, I've done a couple posts looking at kind of the COVID-19 pandemic. And I always try to kind of tie it back to evolution, because biological systems, or evolution, is the harshest critic that we know. And if something exists there, then it has survived multiple rounds of evolutionary forces trying to kill it. And so it has this kind of sense of perseverance. It has this it has some quality that has enabled it to stick around for a while. Like I think in Why We Get Sick, he touches on things like fevers and morning sickness. And yes, those are horrible and unpleasant, but they exist evolutionarily, and there has to be a reason for it. And so understanding that reason just kind of helps us understand a little bit better how we survive, and kind of what we can actually do to improve human health in a way that's not detrimental to other aspects of our human health.
Wonderful. Let's see. You have a bunch of slides, so why don't you go through your slides?
I actually only had 2 more slides aside from that.
Go ahead. Go for it.
Those were just notes that we've pretty much touched on already.
Okay.
So like kind of looking at, I just kind of wanted to bring up things like the skin, and I guess also the oral microbiome, as well as just in general, some of kind of the key characteristics that allow us to work well with bacteria, and that
let me do one thing, because the thing is that I can talk to you for hours. I usually do talk to you for hours, so that's a problem. So I want to correct it. There are 43 people here who will have lots of things to ask. So I want to open it up in a bit, but I want to talk a little bit about your blog and the range of what you have covered. So talk a little bit about the range of what you've written about, what you're interested about.
Okay, so So actually, a lot of what a lot of what kind of drove the initial content of my blog was things that I learned in college. So a couple of I had like one class where all we had to do for that class was just each week we had to read a different paper and then kind of give a talk about it and then analyze it. And then I had another class where I learned about microbial metabolism. And the professor for my microbial metabolism class, kind of at the end of every lecture, would say, okay, we are learning about how bacteria convert, for example, nitrous oxide back into nitrogen gas that is found in the atmosphere. And, you know, we go through all the nuts and bolts of it. And then at the end, he'd say, okay, you know, why do we care about this? Well, it turns out that the bacteria that do that, it's a reaction that needs to be done anaerobically. So in the absence of oxygen, but the bacteria that do it live aerobically in the presence of oxygen. So how do they do it? Well, it turns out they have something like, they have like these little chambers within the bacteria that that are so tightly and hermetically sealed that people are now analyzing the molecules that make up these chambers and trying to use them in industry to create better and tighter adhesive seals. It also turns out that these bacteria generate a molecule called hydrazine in the process of converting ammonia into nitrogen gas. And hydrazine is the primary ingredient of rocket fuel. So, you know, understanding microbiology and bacteriology kind of not only helps us know a system a little bit better, but understanding the nuts and bolts behind it helps us kind of link different fields and see where we can apply this knowledge somewhere in the course of, I guess, humanity that we've built.
Wonderful. Let's do one thing. Let's finish up the story of human beings. The story of what happens to gut biome across a human lifespan. So in the infant, it develops. We talked about it developing in the infant. We've talked about what antibiotics do to it. What else would you like to highlight? What kind of events happen to people's microbiomes?
So diet is one thing that I'd like to highlight. And so I think I'd kind of mentioned like all animals have basically the same metabolism. So our metabolism and the way that it works from glucose onwards is exactly the same as a cow's. However, a cow can live by just eating grass all the time, and humans cannot. Even though I don't know if anyone's tried eating grass, but it's not great. And the reason why cows can do that is because of their gut microbiome. It's because, you know, they have kind of 4 different stomachs and a lot of bacteria that just work at breaking down the complex cellulose and fiber structures of plants into things that are usable for cows. And that the exact same thing happens with humans. And that's kind of one thing that a lot of people don't realize about fiber is kind of beyond its digestive benefits. Fiber, the reason why kind of people encourage eating fiber is because these kind of complex sugars that make up fibrous materials within plants are food for our gut bacteria. So essentially the human diet, a lot of it is kind of secondary feeding, if that makes sense. We don't digest exactly what we eat. And so by eating more fiber, you're conditioning your gut microbiome to kind of have this slower digestion, to get more out of every little bit that you eat. As opposed to kind of the Western diet, which is, you know, simple sugars, processed foods. The problem with those is those go directly through the digestive system and into the bloodstream because your body needs glucose and all you're essentially eating is just straight glucose. So there's no kind of digestion that's required. And so diet, there are a lot of different, actually I see in the chat, David had brought up that fiber is a prebiotic. And that is, that's a key term. Thank you, David, for bringing that up. But So prebiotics are foods that are foods that feed our gut bacteria rather than feed us. And in the case of fiber, by feeding our gut bacteria, we're indirectly feeding us. It just takes longer, and so you feel fuller for longer. And that's why fiber and kind of plant diets are considered, or one of the reasons why fibrous diets and plant-based diets are considered healthier. So as I'd mentioned, the gut microbiome doesn't really change too much in adulthood, barring some major like fecal transplant or long antibiotic course or immunosuppressive course. So there's not much to, I guess, go for, or there's not much to talk about in terms of development of that. And it's more so this idea of maintenance and kind of creating a gut microbiome that is resilient and stable enough to kind of keep your metabolism.
If you wanted to say, okay, what recommendation that most people would agree with, people who are studying the microbiome, what are its implications for your health? If you say, okay, I realize that there is a gut microbiome I want to do the best I can, given that, what kind of general recommendations would people give?
I mean, the two big recommendations that have become more and more commonplace are taking probiotic pills with antibiotics. So probiotic pills are things like lactobacillus, or I think I mentioned some Bifidobacter species, and those are kind of the key commensal drivers of gut microbiome, or of kind of commensal species within the gut microbiome. And so kind of taking those probiotic pills alongside antibiotics helps you kind of fight against antibiotics wiping everything out, and kind of gives your body a bit more of a fighting chance of keeping your gut microbiome resilient over time. So that, and then just eating fiber and kind of more fibrous foods in general.
Wonderful. Let's do one thing. I'm eager to give chance to everybody to ask you questions. So folks, the way we are going to do this is that we are just going to take all the questions first. So we see all the questions, and then we're going to organize all of them. And thanks, Joe. So the book, Joe, thanks for putting both the URLs. The first one for Amazon, Why We Get Sick. It's that book with Ness and Williams. And then he's followed up with a book on good reasons for bad feelings, where he tries to apply the principles of evolution to psychiatry. And he had come to 52 Living Ideas to talk about the book, and that's the YouTube video for it. All right, so folks, go ahead and type exclamation mark if you'd like to ask questions. I'm going to collect all the questions, and then we're going to try to answer as many of them, hopefully all of them. So we're going to start with Jang, followed by David. Jang.
Yeah, this is quite fascinating because recently I actually just bought 2 from Costco last week. This probiotic, because I mentioned earlier, both my son have allergy, especially the younger one. And then we realized he may have ADHD as well. So I joined this nutrition program and we learned a lot of gut health, all this leaking gut. So my question is, and we did this food sensitive test, and we get all this result, and we don't know what to do with it. This leaking gut, then they have this program, you're like, get rid of the food for a week or something. They're just like, the more we learn, the more confused we get, and I know another mom whose kids have autism. He sent me this video I shared in the group, and it's amazing how the woman was able to use food, get rid of the gluten and dairy, then glumia, what I can't remember the name. Then she was able to cure her daughter very dramatically with autism. It's just how powerful the food and, related to our health, and we don't know. And the problem is like, the problem is what we do, it's all this information since what the strategy, like, for example, leaking gut, what do we do with it? Another question I have, one more, very quickly.
Okay.
One more, because because this is so fascinating what you're
I
talking
know
about.
this is a topic of great interest to
This
you.
is
Go
a
ahead.
very different
Go
question.
ahead. Go
I
ahead.
think
Yeah.
So I talked with my son about actually how we go to the other planet to live. And so he actually mentioned how, Mars doesn't fit for the living because of the temperature. So I was wondering, because you were mentioning about, so what you're doing is so fascinating because you could transfer some kind of like bacteria to change the planet, then we can suddenly go there. So that's
Thank you. Thank you. All right. Next up is going to be great questions, Zheng. I'm going to put it as, how do you interpret all the information that you can get about your microbiome to actually come up with a strategy, health strategy? And the second one is about transforming Mars. Next up is going to be, I don't know whether we're going to be able to get to Mars, Zheng, but we will definitely get to health. Next up is going to be David, Joe, Saroj, Deepak, Jack, Olga, Dina, Nancy, and Monique. Okay, so let's start with David. David, what's your question?
Yeah, that's great. Thanks, Shrikant. Thanks for this great talk, listening to this. So I had a question. I know it's sort of a balance between how much we're getting into medicine. I mean, like I say, is a microbiologist, not a medical doctor. I was curious about the analysis of the microbiome in the small intestine versus the large intestine versus the appendix, and specifically about the possibility of the appendix being this repository for restocking a depleted biome, and how much that might has a larger, I mean, if that microbiome matches the microbiome of the intestine, or if that is a repository, as you said, as it gets reduced on, it gets abused by antibiotics or such, that can replenish the microbiome, if that has a broader spectrum of bacteria.
Wonderful, excellent question. I'm going to focus on the appendix part of it, saying, what's the role of microbiome in the appendix. Next up is going to be Joe, followed by Ankur and Saroj. Joe, go ahead.
Yeah, this has been an amazing discussion. So thank you.
It's just beginning, Joe. The question. Look at the questions.
We're canceling the 2 o'clock after this. So, but one of the things that I was wondering is that, antibiotics and some of these things that we've taken, what are the long-term implications as far as how they impact our future mental disorders like dementia or Alzheimer's, and is there a relationship there? Are they able to tie that relationship together? I could ask another question about why costs have come down so much, but I'm pretty sure I know that it's related to computing, but I'll leave that alone.
Thank you. So what are the long-term impacts of probiotics or any regimen that you do?
Antibiotics. Because the antibiotics.
Okay.
So antibiotics would and do they result in mental
Okay, wonderful. Excellent. Thank you. Thank you for correcting. Next up is Ankur, followed by Saroj. Ankur, can you speak out your question so everybody knows?
Yeah, no, it's very fascinating conversation. Thanks for this. I was actually just going to ask something very similar along the lines of what Joe just asked the question about. So yeah, I think so. Mine is already covered. Thanks.
Wonderful. Thank you. Next up is Saroj. Saroj,
yeah, hi. Hello, everyone. So it has been very interesting to learn. So my thing was when you talked about that how the diet makes a kind of a big difference. Mm-hmm. So when you talked about probiotic, how does it help? And how long should someone continue with the probiotics?
Wonderful.
With this.
Thank you. Thank you, Saroj. How exactly does probiotics help, and how do you use it? Next up is going to be Deepak. Deepak, go ahead. Deepak, would you like to? Okay, let me go ahead and ask. Go ahead.
Can you hear me? Okay.
Yes.
Thank you, Shrikant and Agastya. And thank you for the topic. And I had 2-fold question. One is pretty basic. Can you measure your gut health? What are the symptoms of a bad gut biome? And the second is, I'm looking into the terms like intermittent fasting and time restricted eating. So does this impact the gut health? Can positively or negatively? And is there any science behind it? Thank you.
Wonderful. What is bad gut health? And I'll broaden the question. Do diets like intermittent fasting impact the gut biome? Thank you. Next up is going to be Jack, followed by Olga. Jack.
Yeah, wanted to thank you for having this talk. I really appreciate it. So I have like a couple of questions.
Mm-hmm.
One is, I'd imagine that refined sugar and processed foods, they have a direct impact on, say, development of metabolic syndrome, and they cause damage. I would imagine that they'd cause damage to the gut biome, microbiome bacteria. I've heard probiotics, and as is mentioned, probiotics are a good choice for supporting good bacteria. Are there clear-cut foods that have been shown to be doing permanent damage to the gut microbiome that people would not normally consider as harmful? And then my other question is, you mentioned the connection to the brain via the vagus nerve. I've heard the gut as referenced as the second brain. In what ways has it been shown that the gut has an influence on the brain in a healthy individual? And then finally, just to echo, what someone else is saying, is there a specific biomarker for the gut biome that someone could measure through a blood test that someone could take? That's it. Thanks.
Fantastic questions, Jack. Thank you. Thank you. So I'm going to put it first in terms of, are there some foods that do permanent damage, like maybe refined or processed foods, for instance? Second is that what is exactly the brain and gut connection? And the third one is, are there any markers? How do you measure gut health? Next up is going to be Olga, followed by Dina. Olga.
Hi. Thank you for a great presentation. I have actually many questions, but I try to ask only a few. So first, there are some kind of probably recent research that it's not only species of bacteria important, but also strains of bacteria. And some species of bacteria, they say, only survived like in some rural area in South Asia, in some tribes or something. And so like we already lost them. And also there is microbiome, but it's also macrobiome. It's like what you said about forest, about environment that we live in. And it's so altered. For example, there is recent research on rivers in England, and they say there is no clean water anymore in rivers. It's just chemical mix.
Olga, can you summarize your questions very quickly, because we've got many questions and then we have to answer them.
The main question is about species, strains, and micro and macrobiome, biome that altered by pharmacology, by not only like antibiotics, but many other pharmacological medications, like wonderful, even like anything, like antidepressants.
Thank you. Next up is going to be Dina, Nancy, Monique, Sharon, and Dave, Judy and Dave. So Dina, go ahead. Agastya, don't worry. This is common to have these many questions. That's what we always do. So don't worry.
It's fine. Thank you all for your questions. I'm excited to get into this.
Excellent. Dina, go ahead. Okay, next up is going to be Dina. No, Judy, go ahead.
I have two questions. One, does diabetes affect the microbiome? Being diabetic, does it affect the microbiome? And also the second one is, what about colonoscopies? When you have colonoscopies, how do they affect the microbiome system? It seems that I've had several. My mother died of colon cancer, so I'm in this program that I have to have one like every 4 years. And ever since then, my microbiome is I'm allergic to everything, and I can't eat this and I can't eat that. So I mean,
it.
I was just wondering if there's a connection.
Thank you. Next up is going to be Nancy, followed by Monique. Nancy.
Yes, sorry, took me a minute to unmute. Yes, first of all, I want to say I love it when a student, my own student, teaches me.
That's what
by my own student. That's the most wonderful feeling for a teacher.
Nancy, I have to say this. Leonardo used to say, he's a poor student indeed who doesn't exceed his master.
Exactly.
So you're in good company.
So true. Well, so I wanted to know, I thought it was fascinating what you were saying about the oligosaccharides in human milk. But even before that, when you were talking about the gut biome and how important it is to human digestion, so has there been studies on the evolution, the coevolution of the gut biome and human, the different changes in our own body shape and our own, all the evolutionary changes that have happened to humans over time, and how that's connected to the evolution in our gut biome? Because I would assume having the ability to digest food more effectively would be a huge ecological and evolutionary advantage for an early human.
Wonderful. Thank you. Great question about the coevolution of the microbiome and human beings. Next up is going to be Monique, followed by Sharon. Monique.
Hi, thank you for this. My question is regarding whether you can introduce a bacteria to get over certain allergies. So I developed dairy allergies or sensitivities as I got older. And even though I take probiotics now, it's still there. So is there something that clients can do to help?
Excellent. And allergies be cured by manipulating the gut microbiome. Thank you.
Thank you.
Next up is going to be Sharon, followed by Dave. Sharon.
So my question's sort of similar to Monique's. It's more about diet. If you have a vegetarian or vegan diet, does that affect gut biome?
Wonderful. Vegetarian or vegan diet. Excellent. Next up is going to be Dave.
Yeah, great presentation. I will admit to playing outside as a child and eating grass. And been pretty healthy, but I've heard commentary that our children now live in a sterilized environment, and that's why there are so many allergies, because the bacteria don't get into their system. We'd like to comment on that.
Wonderful, wonderful. Excellent. Folks, fantastic questions. Just love the questions. All right, so what we're going to do is that I'm going to organize these questions, and we're going to go kind of look at the more general, large scale questions first, and then more specific questions. The largest, so I'm going to start with the question about coevolution. Agastya, can you talk a little bit about the coevolution of human beings and the gut microbiome?
Yeah, so that's, if I'm kind of completely honest, that's not something that I've studied all that much. But I mean, the microbiome, seeing as, kind of animals have resident microbiomes as well, the microbiome is not kind of unique to humans. And so it does kind of predate humans a little bit in kind of in evolutionary age. However, I mean, kind of going back to what we were saying about, kind of long acting, or like this long digestive process. So looking at like kind of human origins and back before the pre-agricultural revolution, where humans followed kind of a more animalistic diet, where you're eating kind of meats only very rarely based on what you can hunt. And you kind of have to have like the metabolism to sustain like long distance. So human beings I know we're used to like not having as many good physical characteristics as other animals, but human beings are the best long distance runners on the planet. And so that I'd be surprised if the gut microbiome doesn't play into that sort of endurance of being kind of able to run and, you know, just follow animals until they get too tired to fight back. There has to be some kind of link between that and you know, the fact that by eating mostly plants and very few meats, your gut is going to be fuller for longer and your microbiome is going to take a longer time to digest. Beyond that, I'm not 100% sure of studies that have been done. I haven't come across any, but then again, I haven't been looking for them. I'd be happy to follow up with them though.
Wonderful. Thank you, Agastya. I really like the answer. I particularly like the part about the fact that you, in order to understand human beings, and that book, Why We Get Sick, does a fantastic job of thinking along these lines. If you want to understand anything about human beings, you have to look at our evolution. The fact that, you know, okay, we are long distance runners, you know, that's how we operate. That is a very relevant thing to look at. You wanted to say something?
Oh, no.
Okay, sounds good. All right. Next question. So what I've done is that what I do is that I organize the questions by chunk, by topics, and I'm going to combine many of the questions. So the next question is going to be about the brain and gut health. The one after that is going to be about measurement. How do we measure, you know, the health? What exactly is going on? How do you interpret anything? Then we're going to talk about the impact of the diet. Those are the three topics coming up. So Agastya, can you talk a little bit about the connection between the brain and the gut microbiome?
Right. So the connection between the brain and the gut microbiome, which I'm sorry, I forget who had initially asked this question, but they mentioned the vagus nerve. And so that's just kind of big nerve bundle that goes, and it's basically a direct link to the brain. So people have found that the gut microbiome can influence certain, the gut microbiome can influence or has relationship to certain mental health diseases, such as I think I mentioned in the past, autism and schizophrenia. And in fact, they found that there was a study that was done recently on mice where using a mouse model for autism compared to, or I guess compared to wild type normal mice, they found that administration of, it was either a species of lactobacillus or listeria, and I'd have to look that up, but administration of this was able to partially reverse some of the symptoms of autism. And while this doesn't, I mean, I don't want to overstate the results of this because it's not, you know, a study that's done in humans and hasn't really been tested to make sure it has that effect. You know, it does kind of highlight that there is this causal relationship between the gut microbiome and mental health that we still don't completely understand. There have also been studies that have, so there are a couple different hormones that are responsible for when we think about like kind of our gut signaling to the brain, a lot of that is I'm hungry versus I'm full. And so the two hormones that kind of control that are leptin and ghrelin. And ghrelin kind of sends a hunger signal, leptin sends a full signal. And one thing that people have found is that gut microbes tend to grab and utilize ghrelin and leptin that have been released by intestinal epithelial cells. So these are the cells that are the lining of the intestine, right in direct contact with the lumen, which is just the like the space inside the intestine that food passages through and that bacteria live in. And so one thing that has been shown to be really clinically relevant is that gut, depending on what species of gut bacteria you have, and I don't know off the top of my head the exact ones, unfortunately, but you can, those species tend to kind of metabolize these hormones and prevent the signal from getting to other parts of your body. So the microbiota definitely has, it can be a beneficial or kind of detrimental middleman between communication in different systems in the human body.
Wonderful. I want to ask one more. I keep reorganizing these things, the
Yeah, no problem.
questions. And there is one theme, you talked about human evolution. I want to connect to that. In modern times, Dave was pointing out we live in children live in a sterilized environment. We also eat different foods. Like, for example, most of the food that we eat is more processed. The variety of what we eat is much more limited, especially in the West, than what we grew up eating. What is the consequence of the modern, let's say, Western lifestyle of how we eat, how we live on the gut biome as best as you can see it?
So I'm gonna kind of talk a little bit about because I've actually done a very little bit of work in this area. One of the labs that I rotated in works on kind of understanding the gut microbiome as a system. And so one of the things that they're doing is, I'd mentioned, how cheap DNA sequencing is now, but generally when people do kind of dietary surveys, they, you have to write down what you ate, and people aren't going to be honest because they, you're not going to say I ate an entire sleeve of Oreos last night, even if you did, and that directly influences the study. So what people have, or what this lab does is they have this this technique called diet metabarcoding, where you take DNA, isolate DNA from someone's fecal samples, and then just sequence all of the plant and animal DNA you find in there, and get a list of species so you know exactly what they've eaten. And what people have found with looking at diet metabarcoding compared to something known as the Simpson's index, which is an index of it measures species diversity in a system. And what they found is looking at the nutritional content of species versus the Simpson index. As you increase the diversity of your diet, you do have a positive correlation in the increase of nutrients that you get. So more diverse diets tend to be more nutritious. And that's something that you can kind of keep track of as like a measurable for gut health. Another thing that kind of relates to what you were saying, going back to the allergy and like kind of odd, I guess, immune overreact. So allergy it's just an immune overreaction. So without going too deep into the immunology weeds, which I could spend all day doing, there's an arm of the immune system that targets helminths, which are kind of parasitic worms. And in the Western world, we've largely kind of eliminated helminths as a problem, although helminths are still a massive problem in more kind of developing parts of the world, such as Africa, South Asia, and South America. And what we've found is that the same cells that are designed to respond to helminths are the same cells that respond in the case of allergy. So one of those cells, for example, is mast cells, is designed to release a large amount of histamine to break apart and break open a helminth so that smaller cells can get into it. But now the issue is that these same immune cells are starting to recognize things like dust or egg, kind of epitopes within eggs or peanuts, for example, and they're causing these allergic reactions to foods that are totally benign. One thing that people have done to kind of counter this a little bit, there are a couple, one of them directly involves the gut microbiome, is taking advantage of how genetically manipulable bacterial species are. There are companies that are working on, or that have made enzymes that can break up things like tryptophan, for example, which people have, is an amino acid that people have an alert, that some people can't eat due to allergy. But if you have kind of this bacteria as a resident within your gut, or this genetically engineered bacteria as a resident in your gut, it creates an enzyme that can break apart tryptophan that your body doesn't create if you have that allergy. So we can engineer the gut to kind of break apart allergens before they get to a point where they cause allergy. Another thing that people have found important, which kind of goes back to a little bit about what Dave was saying when he was playing in the dirt and eating grass. He was saying that, like, you know, people are just introduced to introduction to a lot of things as a child when your gut microbiome is developing are key. And there is actually a study that was done recently where they looked at peanut allergy incidence across infants in the US versus Israel. And what they found is that peanut allergy is dramatically lower in Israel than it is in the US. And that's because of kind of medical, or between like medical guidance and like available snacks. Like, you feed toddlers Cheerios, let's say, which are just, and the plain ones are just basically just wheat kind of molded into a Cheerio shape. But in Israel, what they give babies as like a snack are these snacks called bambas, they're oats. Yeah, thank you, David. But they give people these snacks called bamba, or I think they're called bambas, I'm not sure. But they're basically peanut puffs. And because these kids are constantly eating peanut puffs, they don't have peanut allergies. Whereas in the US, kids are introduced to peanuts much later. And actually that's led to a recent change in kind of the CDC, or actually, I don't know if it's the CDC, but there's some medical body that has been giving out advice in the US about when to start introducing foods to kids. And they're starting to lower the age at which they're recommending introducing things like peanuts and other tree nuts to kids to kind of see if that will stem some of the allergy problems that have been kind of becoming more and more commonplace.
Wonderful. Now, a tough question. How do we measure gut health? There's just so much, there are all these bacteria, all these complex things. How do we measure it? And how do we conclude from whatever measurement we do about what we need to do for ourselves?
That's definitely a tough one. And so this is kind of where I'm going to put in a caveat where, gut microbiome science is really cool. And there's a lot that we're finding out. And now comes kind of the big step of taking what we find out and translating it into usable products. And that is still very much in development. As to like kind of how to do that outside of a clinical lab setting. Because obviously not everyone has like DNA sequencers sitting on their desks. No, I
want to interrupt you just for a second here. So firstly, I want to say that this is the nature of science, right? Of asking questions. Even when you know that you don't know the answer, you do not have the capacity right now of actually answering them. You're saying that, look, this is a part of me that I need to understand. I understand it only up to some level, and I will keep working at it, and try to figure things out. I'll try to be as cautious, as comprehensive as possible. So this kind of scientific tenor of mind, I think is crucial. And what we have is as a result of it. There are some amazing things that come out of it, and some of the amazing things that come out of it, they are a result of a long preparation, long work that happened before, and then suddenly you have something. So please go on.
Yeah, so I was just going to say, kind of things are kind of coming up in the probiotic field, and there are companies working on systems that I'd mentioned before, where they have their own bioengineered bacteria that you can kind of take, and then if you take other like metabolites that only that bacteria can digest that are still biosafe, you can make sure that bacteria establishes itself within the gut, and then you can start kind of doing what are known as like effector functions. So helping with digestion, acting as kind of a signal or a molecule. So those are kind of still in development, and I can't honestly speak to, I guess, what stage they are or their efficacy, because I don't personally have experience with that. But that's definitely something that's in the works, because once you find, it's just human nature, isn't it, to kind of say, once you find something, your next question is, oh, how can I measure it? Yep. And so that's the kind of, we're right on the cut in kind of that transition phase between we found out what it is and we need to kind of measure it and develop ways that we can actually track it. So
wonderful. Next question was on antibiotics.
Yeah.
What are the long-term impacts of the antibiotics and the way antibiotics have been used in the past? How do you see, when given what you know about gut health, where do you see antibiotics going in the future?
Okay, so one of the one of the things that I just like telling this story because it's cool, that bacteria can do this. One of the things that bacteria can do is known as horizontal gene transfer. So humans are capable of vertical gene transfer, which is genes passing from a parent to a child. Horizontal gene transfer is genes passing from an individual to another individual. So for example, if I had the gene for brown eyes, then I could, if I was capable of horizontal gene transfer, I could just take that gene and then give it to Shrikant, and then Shrikant would have brown eyes. But humans obviously can't do that, but bacteria can. And that's one of the reasons why antibiotic resistance spreads so much and is not species specific, is because you have these antibiotic resistance genes that develop in a certain species. And then you can take, and then those genes just get shunted into other bacterial species almost indiscriminately. And that happens in places of high bacterial content, like I mentioned, the human gut. So I think one of the bigger issues with antibiotics currently is, that is just happening on a massive scale. Anytime antibiotics are being taken. And I also think that, the one other big issue that I mentioned earlier is like a C. diff infection. So what happens is when you give kind of these broad spectrum antibiotics, then you wipe out the resident gut microbiome when you have this kind of empty intestine. But if you look at it, it's a perfectly anaerobic environment. It's going to be rife with food, and it's completely safe. And so it's a great environment for bacteria to colonize. And C. diff is extremely opportunistic in this regard. So it kind of goes in and dominates the gut and causes like gut inflammation and other negative health outcomes. And so the issue with once that kind of establishes itself, there aren't really any antibiotics that can deal with it. And so what you have to do is something known as like a fecal transplant, where you take someone else's poo and then distill all the microbes from it into a pill and then eat it. And that then kind of establish it within your microbiome. I think there are a lot of kind of individualized therapies that are coming up. And I'm kind of excited to see how that plays out, because we're getting better at kind of diagnostic rapid diagnostic tools for understanding which different bacterial species are infecting us. And then there's this technique known as phage therapy, which I have a blog post about that I'm happy to link. But phage therapy is the idea that there are viruses that infect bacteria, which are known as bacteriophage, that are hyper specific to the bacteria that they can infect. And so by administering bacteriophages along with antibiotics, you can kind of get this extremely highly targeted approach where you found the one bacteria that's causing an infection and you go and you target that exact one without disturbing the resident microbiota. And phage therapy is not a new technique. It was used kind of extensively by Russian soldiers in the past, and there's a large institute for phage therapy research in like the country of Georgia. So it's something that has been going for a long time, but the problem is there's kind of a spectrum of specificity and efficacy, if that makes sense. So you can have something like a broad spectrum antibiotic, which is not specific at all, but it's highly effective. I mean, human health broad spectrum antibiotics, because of those, human health has been dramatically improved. And on the other side of it, you have kind of phage therapy, which is highly specific, but if you don't know exactly what you're targeting, then it's not very effective. So you need to kind of find a happy medium, and that's what's going on right now. In terms of that, is finding this happy medium between personalized therapeutics versus broad spectrum therapeutics.
Wonderful. So I'm going to end this meetup in a very interesting way. So I'm going to ask Agastya a question right now. Then I'm going to call upon people to talk about what they got from the meetup. And then finally, I'm going to highlight one additional post by Agastya on his blog. So Agastya, so if you'd like to talk about what you got from the meetup, go ahead and type an exclamation mark in chat. Agastya.
Yes.
We've just touched upon this subject. There is a lot more to know. If somebody wants to know more about this subject, what should they do?
If somebody wants to know more about the subject, follow my blog. That's a good one. There are also, Twitter is a very active space for science, and that's kind of come up recently. And so it's gotten to a point where a lot of individual scientists and a lot of labs have their own Twitter handles where they tweet kind of new things that are coming up, links to papers, things like that. And so I can send Shrikant a list of kind of good places to go from here, and then if and he can send it out to everyone.
Sounds good. So I'm going to post it on the on the meetup page for this. And Rupali, if you could put Agastya's blog in the chat, that would be fantastic. All right, folks. So now, what did you get from this meetup? It's going to be Judith, Evanique, and Joe and Zheng. Judith.
This was an amazing presentation and the questions were awesome. I'm so good. I agree with what I just saw go by in the chat. If you had a discussion like this regularly, I think we could get so much from it. Just thank you so much, Agastya. And I mean, I studied nutrition 40 years ago. And I just, microbiology, biochemistry has just come such a long way. And diet is just so daily critical to every single person. So it's so relevant. I love it. And thank you.
Thank you, Judith. Next up is Evanique.
Thank you, Agastya, and thank you, Shrikant, for having this. I've learned so much, and I am not a science person at all, and I was able to follow it and understand it and really get so much information. I've favorited your blog, Agastya, so I'll be looking at that today. And so this is amazing, and I agree. Like, I would love for you to come back and do another one. And I agree with more nutrition topics because it really is essential to human health. So thank you for what you're doing, Agastya, and thank you for sharing it with us.
Thank you, Evanique.
I'm glad you enjoyed it.
Next up is going to be Joe, followed by Zheng and Rupali. Joe.
Yeah, I mean, there are so many things that I've actually taken from this. I mean, the relationship with the vagus nerve, the example of the mice, and also on your website, just the relationship between, or how costs have come down to be able to run these reports is a very important part of this as well, that we just, we just touched upon that. But there are a number of things. And one of the things that was very interesting is like the idea of fat in our diets. I wanted to touch upon that at some point, but that's a really important thing. I think that, we go with this low fat diet and it's a misnomer. But anyway, there's a lot I learned in addition to everything else. It was a fantastic meetup, and thank you for joining us today.
Thank you for having me.
And please come back. Please come back.
I'd love to.
Wonderful. I guess it looks like lots of people want you to come back. Let's see more here. Zheng, followed by Rupali. Zheng. Zheng, we can't hear you. There is a problem with your sound.
Can you hear me now?
Yes. Go ahead. Yes.
Okay. Yeah, so this is very hard. I was studying this in the last 2 years, nutrition and all this. I'm trying to figure out. I was looking into Chinese medicine. I tried the skin doctor, the specialist, allergist, and I finally get to this conclusion. And you basically confirmed. I learned, it's all the gut health. But the question is like we learn this how to solve the problem, another level.
Thank you. Zheng, you're breaking up. Thank you, Zheng. Thank you.
Thank you.
Next up is Rupali.
So this was very fascinating. One of the things that I notice in school is when I work with children, there are some students who have attention issues, and some of our teachers have done this work where they did an experiment where, they tracked the students' snacks in the morning and breakfast, and they measured when they would have a crash in their ability to sustain the work cycle. And so the different studies you mentioned, Agastya, and the connection between the gut biome and the brain and the food we eat and how we function, I thought that, okay, I got some insights into it. There were many different things. The horizontal gene transfer, I had never heard of that before. So isn't that fascinating? So a lot to learn, and very glad to see, from Agastya doing experiments with Nancy using little petri dishes and growing bacteria or microbes or whatever they were growing together to now at this point, and to see that Nancy is here, it's just very rewarding.
It's very rewarding for me too, because at 1 year old, we were going through the garden, looking at different things that were living in the garden. So I remember that. Folks, my second choice was doing, Agastya has written a whole bunch of blog posts on immunology and how that works. So maybe that's something, Agastya, at some point, would you like to come and do one on immunology?
I would absolutely love that. Immunology is kind of I had mentioned earlier, immunology is like the reason I got into this. And it's still really near and dear to my heart. So I'd love to be able to get a chance to talk about it and kind of share what makes me interested in it.
Wonderful. So for the finale, you've written a blog post. Your latest blog post is on how epidemics end. Could you talk a little bit about that? And then we'll.
Sure. So this is going to go back to evolution, believe it or not. But kind of it's kind of something that I've been thinking about recently is, pandemics aren't lasting. I mean, there have been pandemics in history, they've come to an end, and, we've had long periods of time between pandemics. So that kind of led me to thinking, how do pandemics end? And what makes them become a part of our life? And how does that kind of relate to the current pandemic? Because right now, frankly, it seems hopeless because every couple months there's some new variant and a new wave coming up. And so I was kind of looking at some statistics with Omicron, and in what I brought up in that post is kind of an example in rabbits. So in Australia, European colonizers basically who'd gone and who traveled from Europe to Australia when Australia was a British colony, brought European rabbits to Australia so they could continue rabbit hunting like they did in England. And these rabbits became an invasive species and caused, there are hundreds of millions of rabbits at one point by the 1950s running around Australia. And so what they did to kind of cull these rabbits is introduce a virus called myxoma, which is a rabbit virus that naturally circulates within South American rabbits. And so because it causes very mild symptoms, but is highly contagious in South American rabbits, but it causes high rates of mortality in European rabbits. And so what they did is they introduced this virus to the European rabbits in Australia, and the European rabbit population in Australia decreased by 99% as a result. But then what happened is the virus kind of co-evolves along with its host. So a virus, like any other force of nature, wants to propagate. And the way that viruses can't propagate on their own, by definition, they're not capable of reproduction. So the way that they do that is by hijacking reproductive, the kind of like cellular systems in other animals. And so if a virus kind of infects a host and then multiplies and then kills its host before it can infect another, then the virus also dies. And so is unable to propagate. So what ends up happening is over time, diseases tend to become less virulent and cause less disease in an individual so that they can propagate more. And as a result, they become more contagious. And so that's kind of what we've seen with the Omicron variant relative to other previous variants, is that the Omicron variant causes much milder disease, especially in individuals who are fully vaccinated and boosted, in that it barely reaches the lungs, whereas Delta, the Delta variant completely shredded people's lungs. And even though breakthrough infections are very common now with Omicron, the virus itself kind of just hangs out in the upper respiratory tract, doesn't get down into the lungs, and as a result is able to be cleared quicker and isn't able to kind of take hold and create massive tissue damage. And so that kind of mirrors what we've seen in the past, where this viruses get less and less severe and more contagious. And that's in fact actually what happened with the influenza epidemic of 1918, is that the influenza virus, after a few years, became this kind of, the dominant variant became something that was much more contagious and much less virulent. And then its virulence just kind of kept decreasing over the past, 80, 100 years, into the flu virus that we know now, which is seasonal and fairly benign, especially if you've gotten a flu shot and you don't have any existing health comorbidities. And so that's kind of, it's, while Omicron doesn't necessarily doesn't signal the end, and there's no way of knowing whether or not there are going to be more variants, it's definitely it definitely represents a step kind of in the right direction historically, what we've seen with pandemics.
Wonderful, wonderful. Agastya, this is such a pleasure. So let's do one last thing. Let's give a preview of immunology. So tell me a little bit about immunology. Just get people excited about reading about immunology. We'll go ahead. Rupali, if you could search for the immunology post on Agastya's blog and put it in the chat so people can read it beforehand and prepare for it. So say a little bit about maybe 2-3 minutes on immunology.
Sure. So immunology is really, really interesting to me. You'll find this on my blog as well. I've split up immunology into 4 different blog posts because it's way too much to talk about in just one. And the immune system has, I guess, is kind of divides the pathogens that it targets into different categories. And so the way that I kind of think about it is there's one category for intracellular pathogens. So these are viruses and small bacteria and parasites that actually go and live inside individual cells. And then it has one category targeting extracellular bacteria. So these are bacteria like E. coli or Pseudomonas or Staphylococcus that cause biofilms outside of cells and larger scale tissue damage. And then you have this kind of third category, which targets helminths that I mentioned earlier. That's also responsible for kind of allergy and asthma. And this immune response is designed to target multicellular organisms. So how do you take down a worm that is 56 times the size of a single white blood cell? And that this immune response is kind of designed to coordinate that, this like swarming attack. And then you kind of have another arm of it, which is regulation of the immune system. How do you dial it back? And so what fascinates me is just looking at how all of these systems work exactly as they're intended to. And often because of kind of a combination of our better understanding of human health and what Dave had mentioned earlier with the what's known as the hygiene hypothesis, is that we're getting too clean. Even though those like the immune response can be kind of horrific when it goes wrong, I just kind of really like understanding how the immune system works and what is the reason for some of the things like autoimmunity or asthma that we're seeing, and can we use those the basic reasons to manipulate the biology to our advantage again.
Wonderful. All right, Agastya, this was just delightful. And yes. Let's talk about when you want to be back talking about immunology, and we will go ahead and schedule that, folks.
Sounds great.
Go ahead.
I just wanted to thank everyone again. It's a pleasure to be here, and you all had some really great questions. And I'm sorry we couldn't get to all of them, but the ones that I didn't know answers for, I'll reach out to Shrikant and hopefully provide some follow-up.
Sure, and they can like your blog, and they can contact you through your blog also, right?
Yes. Okay. Yeah.
So folks, Go ahead.
I was just going to say, there's like a contact form on my blog, and I check that really regularly, and I try to post every couple of weeks or so. So hopefully you enjoy reading it.
Wonderful. All right, folks. So let me just tell you what's coming up at 2 o'clock. I'm going to be talking to a much younger friend. I've known him only for a year or so. He has more maturity than 1 year would suggest, probably because of the reason what he's going to be talking about. And that's on stoicism and what stoicism can do for you. So that's going to be Joe talking at 2 o'clock. And then at 3:30, we are going to be talking about the concept of center and periphery, that we actually independently ran into the same concept in Gita, the Bible meetup, and the Tao Te Ching in the same week, last week. So I had to look at, and it's the same concept in the Stoicism. It's the core idea of dichotomy of control. The same concept comes up. So we'll be talking about that concept. That's going to be at 3:30. And then at 8 p.m. today, we're going to be talking about talking to Jason and Amon on their experience of translating Tao Te Ching. All right, folks. So thank you very much. And Agastya, always a delight talking to you. I think, I always enjoy talking to you, and we talk a lot. And it's actually wonderful to be talking to you in midst of all these friends. And I really, really like the questions from everybody, by the way.
Yes.
Because what happens is that it's just the range of questions is incredible. And, science is more about questions than answers, because the moment you answer a question, you go to the next question. That's what science is about. So thank you very much. Go ahead, Agastya.
No, I was just going to say, that was one of the things that I wrote in a blog post once. Is like, I think of science as the Greek mythological Hydra, if you're familiar. The idea is it's this kind of multi-headed monster, and whenever you cut off one of its heads, two of its heads take its place. And so it's definitely something where you answer a question, and then all that does is it leads to more questions.
Wonderful. All right. Thank you, Agastya.
Thank you.
See you soon.
Have a good weekend, everyone.